Today, we are tackling, I think, the most fundamental question in all of medicine. How does a drug actually work?
Right.
We've talked about how they get around the body, that's pharmacokinetics, but now we're diving into the real action, the molecular choreography that happens when that drug molecule finally meets its target.
This is the moment. This is where all that chemistry and biology translates into a clinical effect. This is the study of pharmacodynamics.
And it is. It's just critical for understanding medicine at that deep level.
It is. We're not just, listing side effects. We are going to explain the entire mechanism, the full action to effect sequence.
We'll get into the relationship between the dose you take and the response you get.
And even what happens when you start mixing drugs together.
Yeah.
How they can alter each other's actions. It's a huge field.
Our sources give us this incredible blueprint of molecular action, and that's our mission for this deep dive, to unpack it. We're going to start with some of the, let's say, simpler mechanisms, like blocking enzymes.
The molecular roadblocks.
Exactly. And we'll build all the way up to the incredibly complex world of cell signaling, from responses that happen in a millisecond to ones that take days to develop.
By the end of this, you should have a really solid framework for understanding how drugs are targeted and, the rules that govern whether a drug is potent or effective.
So when a drug gets into the body, I think most people imagine it immediately finds something called a receptor.
That's the common picture, yeah.
But so many important drugs, both ancient ones and brand new ones, they target things that aren't receptors in that really strict sense.
That's right. So we should start there with the non-receptor mechanisms. And the first, most foundational one is enzyme inhibition.
The molecular roadblock, as you said?
Exactly. Enzymes are the cell's factory workers. They're a biological catalyst that speed up all the essential reactions you need to live.
So if you can block one of those workers...
you can stop a disease process in its tracks.
Yeah.
Now, we should mention you can have really non-selective inhibitors, things like heavy metals or strong acids. They just
destroy everything.
They denature all the proteins. They inhibit all the enzymes. It's just molecular chaos.
Yeah, highly toxic, I'd imagine.
Oh, absolutely. The real art of pharmacology is in selective inhibition. And we can break that down into two main types.
Okay, so let's start with the first one. Competitive inhibition. This is the equilibrium type.
Right. And your analogy earlier of a molecular foot race is perfect.
Okay.
In this case, the drug, the inhibitor, it's designed to structurally look a lot like the normal molecule, the substrate, that the enzyme is supposed to work on.
So they're both competing for the same parking spot.
The exact same spot, the enzyme's active site or catalytic binding site. And crucially, this binding is reversible. It's a constant battle back and forth, which is why we call it the equilibrium type.
Okay, so if it's a competition, how does that actually play out kinetically? What changes about how fast the enzyme can work?
This is the key. Because it's a reversible competition, if you just keep adding more and more of the normal substrate, eventually the substrate will win.
It just overwhelms the inhibitor by sheer numbers.
Exactly. Which means the enzyme can still eventually reach its absolute maximum speed. It's Vmax. So with competitive inhibition, the Vmax remains unchanged.
But you had to add a lot more substrate to get there. The concentration needed to get going has changed.
Precisely. You've increased what we call the Km. The Km is the substrate concentration you need to get the enzyme working at half of its maximum speed. Because you need more substrate to fight off the drug, the Km goes up.
That's the fingerprint of a competitive inhibitor. Vmax stays the same, Km goes up.
That's the takeaway. And you see this principle all over medicine.
Give us some of the big examples.
Okay, think about cholinesterase. It's an enzyme that breaks down a neurotransmitter. Drugs like phytostigmine and neostigmine competitively inhibit it.
So the neurotransmitter sticks around longer.
Exactly. Or in psychiatry, moxglobamide is a competitive inhibitor of an enzyme called MAOA.
And for gout?
A classic example. Allopurinol. It competitively inhibits an enzyme called xanthine oxidase, which stops the body from overproducing uric acid.
And probably the most common one people have heard of are the ACE inhibitors.
Absolutely. Captopril and all the others in that class are competitive inhibitors of the angiotensin-converting enzyme. It's a cornerstone of blood pressure management.
Okay, so that's the foot race. Let's pivot to the second type, non-competitive inhibition. If the first one is fighting for the front door, what's this one doing?
This one is using the side door. It binds to what we call an allosteric site.
So a different location on the enzyme entirely.
Right. It's adjacent to the active site, but it's not the active site itself. So the inhibitor isn't blocking the substrate from binding.
Then what is it doing?
It's sabotaging the machine. When it binds to that allosteric site, it causes the whole enzyme to change its shape. It deforms it.
Ah, so the substrate can still get in the door, but the machinery inside is broken.
Perfectly put. The enzyme's catalytic property is just reduced. It can't do its job as efficiently anymore.
And because the enzyme itself is physically altered, I'm guessing that flooding the system with more substrate isn't going to help.
It does nothing. The enzyme is fundamentally damaged. So the readiness of the enzyme to bind the substrate, the Km that's unchanged,
So it's top speed.
its top speed, the Vmax, is reduced. No matter how much substrate you throw at it, the broken machine can only work so fast.
So there's the other fingerprint. Non-competitive, Km is unchanged, Vmax is reduced.
That's the core distinction. And again, we have huge clinical examples. A drug called acetazolamide non-competitively inhibits carbonic anhydrase.
And the big one for acid reflux.
Omeprazole, one of the most prescribed drugs in the world. It is a classic non-competitive inhibitor of the proton pump in the stomach, the H plus K plus AT pace.
What about something like sildenafil?
Also a non-competitive mechanism. It inhibits an enzyme called phosphodiesterase 5, which reduces its ability to break down another molecule.
Before we move on from enzymes, the sources mention a couple of fascinating cases that sort of blur these lines. Methotrexate is one.
Yes. Methotrexate is a powerful chemotherapy drug. It inhibits an enzyme called dihydrofolate reductase. Now, you could argue about the exact mechanism, but what makes it so devastating isn't just how it binds, but how tightly.
It has an incredible affinity.
Incredible is the word. It binds to the enzyme with an affinity that is 50,000 times higher than the enzyme's natural substrate.
50,000 times.
Yeah. I mean, at that point, it's not a competition anymore. It's a complete takeover. Even if the bond is technically reversible, biologically, it's so tight that it might as well be permanent.
Wow. And then you have something even more permanent like organophosphates.
Right. These are often in pesticides, and they're incredibly potent toxins because they react covalently with the active site of cholinesterase.
And covalent bond that's a permanent chemical bond.
It's one of the strongest bonds there is. Once that organophosphate binds, that enzyme molecule is functionally dead. It is completely irreversible. The body has to physically synthesize a brand new enzyme to replace it.
So that shows how the actual chemistry of the bond dictates the entire biological outcome.
Absolutely. The kinetics, the severity, all of it.
Okay, so we've looked inside the cell at the machinery. Let's move to the cell's borders now. Our second non-receptor target is ion channels.
The gatekeepers of the cell membrane.
Right. These are the molecular pores that control which charged ions, sodium, potassium, calcium chloride, get to pass in or out.
And this flow of ions is everything for excitable tissues. It's what generates electrical impulses in nerves, in the heart, in muscle.
So if you can control those gates,
You can control how a nerve fires or how a heart contracts. It's incredibly powerful.
and drugs can do this directly, which is what we're talking about now, or indirectly through a receptor, which we'll get to. So what's the direct action?
It's basically acting like a physical plug or sometimes a modulator that changes how the gate opens and closes.
A lot of heart medications work this way, right?
A huge number of them.
Yeah.
Antiarrhythmic drugs like quinidine and amiodarone, for instance, they physically block a specific type of potassium channel in the heart.
By blocking potassium from leaving, what does that do?
It changes the timing of the heart's electrical cycle. It prolongs what's called the action potential, which can help reset an irregular rhythm. They are literally retuning the heart's electrical clock.
What about for blood pressure?
A whole class of drugs called calcium channel blockers, like nifedipine. They block a specific type of voltage-sensitive calcium channel.
And calcium is needed for muscle contraction.
Right, so if you block calcium from getting into the smooth muscle cells in your blood vessels, those muscles relax, the vessels dilate, and your blood pressure goes down.
The sources also mention some really specialized channels linked more to metabolism.
Yeah, this shows the real subtlety of pharmacology. There are drugs like nicorandil for angina or the sulfonylurea drugs used in diabetes. They target something called ATP-sensitive potassium channels.
So these channels can sense the energy level of the cell.
That's exactly it. They link the cell's metabolic state directly to its electrical excitability. In the pancreas, for example, closing these channels is the trigger that causes the cell to release insulin.
And of course, local anesthetics.
They're a perfect example. They work on voltage-gated sodium channels along a nerve axon. By blocking those channels, they physically prevent the electrical impulse, the action potential, from traveling down the nerve.
Which is why you just stop feeling anything. The signal can't get through.
The line is dead.
Okay, so we've blocked the enzymes, we've plugged the channels. The third major non-receptor target involves the cell's logistics team, The transporters.
Right, the traffic controllers. These are carrier proteins that are responsible for moving specific molecules across the cell membrane.
Things like nutrients, neurotransmitters.
Exactly. Sometimes they just help a molecule to fuse down its concentration gradient, but often, and this is key, they use metabolic energy to actively pump molecules against their concentration gradient.
So they're like little molecular cranes actively managing the cell's inventory. How do drugs mess with that?
The most common mechanism is to inhibit the transporter's function. You jam the crane.
What's the effect of that?
Well, the transporter's job is often to clean up, to clear a molecule from the space outside the cell. If you jam the cleanup crew, that molecule, say a neurotransmitter, is left lingering in the synapse for much longer.
And that intensifies its effect.
Hugely. This is the entire basis for many of our most important neurological and psychiatric drugs.
This is how most antidepressants work, right?
It's how most of them work, and it's also how illicit drugs like cocaine work. For example, the drug disipramine, or cocaine itself, they work by blocking the transporter responsible for the reuptake of noradrenaline.
The norepinephrine transporter, or NET.
Right. By blocking MET, you leave more noradrenaline in the synaptic cleft, which enhances that particular neural signal.
So the drug itself isn't activating anything. It's just stopping the natural signal from being turned off as quickly.
That's the critical concept. You're not stimulating a target, you're inhibiting the cleanup process. Look at the SSRIs, the selective serotonin reuptake inhibitors, like fluoxetine.
Their name says it all.
It really does. They specifically inhibit the serotonin transporter, SERT. By blocking SERT, you amplify the effect of the serotonin that your own brain has already released. It just shows that sometimes you don't have to push the accelerator to get a big effect, you just have to take your foot off the brake.
All right, we've laid the groundwork with enzymes, channels, and transporters. Now we have to move into the heart of modern pharmacology, receptors. But first, we need to be really specific about what we mean by receptor.
Yes, this is so important for any learner. The term gets used very broadly, but in pharmacology, it has a very strict definition.
Okay, what is it?
A receptor is a macromolecule, usually a protein, located on the cell surface or inside the cell. Its job is twofold. First, to recognize a specific signal molecule or drug.
And second,
to initiate a response. And here's the key part. The receptor itself has no other function.
it doesn't catalyze a reaction like an enzyme or move an ion like a channel?
No, it's an ignition switch. It senses the ligand, it changes its shape, and that conformational change is the entire signal. The receptor's job is done at that point. It's translated the external binding event into an internal one.
It's the ignition, not the engine.
Perfect. If the drug's target has a pre-existing job, like catalysis or transport, we tend to classify it as a non-receptor target, even though it is technically a molecule that binds a ligand.
Okay, so with that strict definition in mind, we need to get the language right. Agonist, antagonist, partial agonist. These are the words you hear all the time.
They are the absolute vocabulary of this field. Let's start with the most straightforward, an agonist.
This is the activator.
Yes. It's a drug that produces an effect that's similar to the body's own natural signal molecule. It fits the lock, it turns the key all the way, and it initiates the full maximum response.
And its natural enemy is the antagonist.
The antagonist. It prevents the agonist from working. The crucial thing to understand about an antagonist is that it has affinity. It can bind to the receptor very well, but it has zero efficacy.
Yeah. So it fits in the keyhole, but it can't turn the lock.
Exactly. It just sits there, blocking the keyhole so the real key, the agonist, can't get in. It has no effect of its own.
All right, now for the more nuanced one, the partial agonist.
Right. The partial agonist is interesting. It has high affinity, so it binds really well. But even if it occupies every single available receptor on every cell, it can only produce a submaximal effect.
It's a key that fits, but it can only turn the lock halfway.
That's a great way to think about it. It gives you a built-in ceiling effect.
We mentioned buprenorphine earlier for opioid addiction. This is why it's so useful, right?
It is the perfect clinical example. It binds to the new opioid receptor and provides enough stimulation to stop withdrawal and cravings. But its submaximal effect means the risk of dangerous respiratory depression is dramatically lower than a full agonist like heroin or fentanyl.
It gives you a safety margin.
It does. Now, the trickiest one for most people to grasp is the inverse agonist.
Because it sounds like a paradox. It activates a receptor to produce an effect in the opposite direction. How can that be?
It only makes sense if you let go of the idea that receptors are totally silent until a drug comes along. Some receptors have what we call constitutive activation.
Meaning they're always on, just a little bit.
Exactly. Even with no drug or natural molecule around, they're spontaneously active, generating a low-level basal signal, a constant hum.
Okay, so the system isn't off, it's just idling.
Right. An inverse agonist binds to that receptor and doesn't just block it, it actively forces it into a truly inactive state. It shuts down that basal hum. So it produces an effect that is the opposite of what the natural agonist would do.
And we see this with the benzodiazepine receptor system.
We do. It's a key example of where this concept is clinically relevant.
And finally, the big umbrella term that covers all of these, ligand.
Ligand simply comes from the Latin to bind. It's any molecule that attaches to a particular site. So agonists, antagonists, partial agonists, inverse agonists, they're all ligands.
The key distinction being that ligand only implies affinity, the ability to bind.
But it says nothing about efficacy, the ability to actually produce an effect.
To really cement that difference between affinity and efficacy, we have to go back to the historical foundation, to Clark's receptor occupation theory from 1937.
This is still the bedrock of how we quantify drug action. Clark's idea was simple but powerful. The effect of a drug is directly proportional to the number of receptors that it occupies.
The more receptors you have a drug bound to, the bigger the effect.
That was the idea. And from this simple chemical reaction, drug plus receptor forms a complex, which leads to an effect. We get two completely independent properties of a drug.
Let's start with the first one, affinity.
Affinity is all about binding strength. How sticky is the drug? We define it by the drug concentration needed to get a certain number of those drug receptor complexes formed.
So high affinity means you don't need much drug to get a lot of binding.
Exactly. Mathematically, it's described by the dissociation constant, KD. A low KD means high affinity.
And the shape of the molecule is everything here. The source gives a fantastic example of what's called stereoselectivity.
Yes, the mirror image problem. The drug propranolol exists in two forms that are mirror images of each other. The L-isomer is 100 times more potent at blocking beta receptors than the diisomer.
100 times. But they're chemically almost identical.
They are. But the receptor is a complex three-dimensional pocket. And only the isomer has the perfect shape to fit snugly into that pocket and achieve the strongest binding. That huge difference in potency is purely a matter of affinity.
So affinity is how well it docks. The second property, intrinsic activity or efficacy, is what happens next.
Efficacy is what really matters for a drug to be useful. It's the drug's ability, once it's bound, to actually make the receptor do something, to induce that functional change and create a biological response. We usually scale it from 0 to 1.
So a full agonist has high affinity and an intrinsic activity of 1.
A competitive antagonist has affinity, but its intrinsic activity is 0. It binds, but does nothing.
And a partial agonist has affinity and an intrinsic activity somewhere between 0 and 1.
Exactly. And they are completely separate properties. A drug can bind like superglue, but have 0 effect.
This framework led to a really surprising discovery, though, the idea of spare receptors.
Yes. Clark's original theory kind of implied that you need to occupy 100% of the receptors to get 100% of the effect. But we now know that's not true at all.
What did we find?
For many powerful full agonists, they can trigger the absolute maximal cellular response while occupying less than 1% of the available receptors.
Less than 1%. So, why does the cell bother making the other 99%? What's the point of this huge reserve?
But point to a massive signal amplification system downstream. One single drug receptor binding event can set off a huge cascade inside the cell. Having this receptor reserve means the system is very sensitive and also very robust.
Robust how?
Well, imagine a disease state starts to degrade or reduce the number of receptors on your cells. With this massive reserve, you could lose a huge number of them and still be able to generate a maximal response when you need to. You have a huge safety buffer.
Okay, the occupation theory is great for explaining affinity and efficacy, but as you said, the inverse agonist really requires a more dynamic model. That brings us to the two-state receptor model.
Right. This model accepts that the receptor isn't just a static waiting station. It's constantly flickering between at least two different shapes or states.
An active state, Ra, and an inactive state, Re.
Correct. And in the absence of any drug, there's an equilibrium between these two states, which usually heavily favors the inactive, Ra state.
Unless there's that constitutive activation we talked about where a few are always in the Ra state.
Exactly. Now, the drug's effect is all about which of these two states it prefers to bind to. A standard agonist has a high affinity for the active Ra state.
So when it binds, it traps the receptor in that active shape, pulling the whole equilibrium over to the raw side.
You've got it. It stabilizes the active conformation. Now, what about a neutral antagonist?
If it's truly neutral, I guess it would have to bind equally to both the active and the inactive states.
Perfect. It has no preference. So it doesn't shift the equilibrium at all. It just gets in the way of other molecules that do have a preference.
And now the inverse agonist makes perfect sense. It must preferentially bind to the inactive state,
Yes. By binding to and stabilizing the inactive state, it actively pulls receptors out of the active pool, which reduces that basal constitutive activity. It doesn't just block, it actively quiets the system down. This dynamic view is really essential for understanding how modern drugs are designed. #, none 3. The action sequence and major receptor types.
So we've got the rules of engagement. Now let's look at the actual gameplay, the full sequence from binding to response. We should clarify the difference between drug action and drug effect.
It's a critical distinction. The drug action is the immediate initial event. It's the binding. It's the conformational change. It's instantaneous.
And the drug effect is the final outcome you can measure.
Yes, the change in your heart rate, the relaxation of a muscle, the relief from pain, that's the effect.
And the gap between the action and the effect is filled by the transducer mechanism.
Exactly. This is the whole intracellular signaling cascade that amplifies and propagates that initial binding event. And understanding these transducer mechanisms lets us classify the major families of receptors, usually based on their structure and their speed.
Let's start with the biggest and most important family, the G-protein coupled receptors, or GPCRs.
The 7 transmembrane receptors, they're involved in just about everything. Vision, smell, hormonal control, neurotransmission. Their structure is iconic.
It's a single protein chain that snakes across the cell membrane 7 times.
It does. It creates this barrel-like structure with a pocket in the middle where the agonist binds. And on the inside of the cell, it's linked up to its partner, the G protein.
Okay, walk us through the sequence. An agonist binds in that outer pocket. Then what happens?
That binding causes the receptor to change shape, especially on its intracellular loops. This new shape allows it to grab onto its G protein partner.
And this is where the switch happens.
This is the switch. The G protein, which is normally holding a molecule called GDP, lets go of the GDP and picks up a GDP instead.
And grabbing that GTP is what activates it.
It's the activation signal. The G protein then splits into 2 pieces, an alpha subunit and a beta-gamma dimer. One or both of these activated pieces then travels away from the receptor to find its downstream target, which is usually an enzyme or an ion channel.
And this whole process, how long does it take?
It's on the order of seconds, which is fast, but not instantaneous.
What turns the signal off? How does it reset?
The alpha subunit is clever. It has its own built-in timer. It's an enzyme itself, a GTPase, and it slowly breaks that GTP back down to GDP.
So once the GTP is gone, it becomes inactive again.
And it floats back to rejoin the beta-gamma dimer and wait by the receptor for the next signal. It's a beautiful self-resetting system.
Now, everything depends on which G protein is actually involved. The sources break them down into Gs, G, and G.
Right, let's start with Gs for stimulatory. The alpha subunit, when it's activated, goes and turns on an enzyme called adenyl cyclase.
And adenyl cyclase makes the famous second messenger, cyclic AMP or CAMP.
Yes. CMP levels go up, and that activates another enzyme called protein kinase A, or PKA. PKA then goes around the cell phosphorylating things, which is the ultimate cause of the effect, like the increased heart rate you get when adrenaline hits your beta-adrenergic receptors.
So G's is the accelerator pedal working through CMP. What about its opposite, Guy?
G is for inhibitory. The alpha subunit does the opposite. It inhibits adenyl cyclase, so GMP levels go down.
It's the brake pedal.
It is. But G can also have a more direct effect. It can directly open certain potassium channels,
letting positive potassium ions leak out of the cell,
which makes the cell hyperpolarized or harder to excite. This is exactly how acetylcholine slows down the heart when it acts on muscarinic M2 receptors.
Okay, so G's and G are the yin and yang of the CAMP pathway. What does the third one, GQ, do?
GO or GQ uses a totally different system. When it's activated, it turns on an enzyme called phospholipase C, or PLC.
And PLC's job is to
chop up a membrane lipid into 2 new second messengers, IP3 and DIG.
2 for the price of 1.
Pretty much. DIG stays in the membrane and activates another kinase, protein kinase C. But IP3 is the big one. It diffuses into the cell and triggers the release of huge amounts of stored intracellular calcium.
And that calcium burst is a massive signal for all sorts of things.
Muscle contraction, neurotransmitter release, secretion from glands.
Yeah.
It's a fundamental cellular signal.
What's amazing is that there are thousands of different GPCRs, but only a handful of these G proteins to go around.
That's the concept of pleiotropy, or coupling diversity. This limited set of tools, Gs, G, has to be shared between many, many different receptors.
So different drugs for different conditions might actually funnel down into the exact same intracellular pathway.
They often do. The dopamine D2 receptor and the muscarinic M2 receptor both use G. The beta-adrenergic receptor uses G. This shared machinery is often the reason for drug side effects, because activating the pathway you want can inadvertently trigger another process that uses the same G protein.
Okay, from a response that takes seconds, let's go to the absolute speed demons of the receptor world. Ion channel receptors, also known as ligand-gated channels.
These are the direct responders. The receptor is the channel.
What's their structure like?
They're made of multiple protein subunits, usually 5, arranged in a circle like the staves of a barrel, to form a central pore that goes right through the membrane.
So when the agonist, the ligand, binds,
it causes an immediate conformational change that physically opens that pore. It's a direct mechanical action.
and ions just flood through.
Down their electrochemical gradient. And because there are no second messengers, no enzymes to activate, the response is
Practically instantaneous.
milliseconds. This is the only way you can get the speed needed for things like synaptic transmission in the brain.
What are the classic examples here?
The nicotinic cholinergic receptor is a great one. Acetylcholine binds, the channel opens, sodium rushes in, and the cell is excited. That's how your muscles get the signal to contract.
And on the inhibitory side?
The GABA and glycine receptors. When their respective neurotransmitters bind, they typically open a channel for chloride ions to enter the cell.
That negative charge makes the neuron harder to fire.
It dampens the signal. And then, of course, you have the NMDA and AMPA receptors, which are absolutely critical for learning and memory.
The sources also mention the benzodiazepine receptor as an interesting case here.
Yes, it's a perfect example of allosteric modulation on an ion channel. Drugs like diazepam don't bind to the main GABA binding site. They bind to a separate allosteric site on the receptor complex.
And what does that do?
It doesn't open the channel by itself, but when it's bound, it makes the GABA molecule much, much more effective at opening the channel when it binds. It's an enhancer, a booster for the natural inhibitory signal.
Okay, let's slow things down again. Moving to a time scale of minutes to hours. The next family is the transmembrane enzyme-linked receptors.
Right, so here the receptor and the enzyme are fused into one single molecule.
What's the structure?
It's typically a single protein chain that passes through the membrane just once. It has a big domain on the outside to bind the ligand, and a big domain on the inside that has intrinsic enzymatic activity.
And that activity is usually a protein kinase.
A specific type, a tyrosine kinase, is the most common.
So how does binding the hormone on the outside turn on the enzyme on the inside?
The key mechanism here is dimerization.
Two receptors have to find each other and pair up.
Exactly. The hormone binding causes two of these receptor molecules to come together and form a dimer. That physical pairing is what activates the kinase domain on the inside.
And what does the active kinase do first?
It phosphorylates itself. It's called autophosphorylation. It adds phosphate groups to specific tyrosine residues on its own intracellular tail.
And those phosphorylated tyrosines then become like a landing pad.
A docking station, exactly. Other signaling proteins inside the cell that have a specific domain called an SH2 domain can now come and bind to those phosphorylated sites.
And that kicks off a whole cascade.
A complex, multi-step phosphorylation cascade that ultimately controls things like cell growth, differentiation, and metabolism. Because it involves all these enzymatic steps, the response time is in minutes to hours.
And this is the system used by some of our most important systemic hormones.
Absolutely. Insulin's the classic example, but also epidermal growth factor, nerve growth factor, they all use this tyrosine kinase mechanism.
Is there another type of enzyme that can be in this family?
There is. Sometimes the intracellular enzyme isn't a kinase, but a guanylyl cyclase.
Like the receptor for atrial natriuretic peptide, ANP.
That's the one. When AMP binds, the intrinsic GC domain gets activated and starts making cGMP, which is another second messenger that mediates things like vasodilation. The principle is the same. Binding activates a built-in enzyme.
Okay, the fourth category is a close cousin to that last one, the transmembrane JAK-STAT binding receptors.
Very similar, but with one crucial difference. These receptors have no enzyme of their own.
They're enzymatically inert.
Completely. They are entirely dependent on recruiting a separate free-floating enzyme from the cytoplasm to do the work.
So the ligand binds, the receptors still dimerize, but then they have to call for backup.
They do. The dimerization changes the shape of the intracellular domain, creating a docking site for a family of cytosolic tyrosine kinases called JAKs, or Janus kinases.
The JAKs are the hired help.
They are. The receptor recruits them, and once they're docked, the JAKs get activated. They phosphorylate each other, and then they phosphorylate the tyrosine residues on the receptor itself.
So the JAKs create the phosphorylated landing pad that the other receptor type created for itself.
Exactly. And once that landing pad is built, another group of proteins can dock there. These are called STATs.
Signal transducers and activators of transcription.
Right. The STATs dock on the receptor, get phosphorylated by the JAKs, and then they do something remarkable.
What's that?
They let go of the receptor, pair up with another STAT, and the STAT dimer travels directly into the cell nucleus to act as a transcription factor, turning genes on or off.
Wow. So this pathway is a direct line from the cell surface to the genome.
It is. It's heavily used in the immune system and for growth regulation. Cytokines, growth hormone, interferons, they all use the JAK-STAT pathway.
That's a perfect transition to our final and slowest receptor category. The receptors regulating gene expression, also called nuclear receptors.
Now we're talking about effects that take hours to even start and can last for days.
And the architecture is completely different. These aren't on the cell surface at all.
No, these are intracellular proteins. They're soluble, just floating around inside the cell, either in the cytoplasm or already inside the nucleus.
Which tells you immediately that the drugs that target them have to be able to get inside the cell.
They have to be lipid soluble to pass through the cell membrane. We're talking about all the steroid hormones, thyroid hormones, and vitamins like A and D.
If these receptors are just floating around inside, what stops them from being active all the time?
They're kept in an inactive state by being bound to chaperone proteins, most famously one called heat shock protein 90, or HSP90.
So the HSP90 is like a bodyguard, keeping the receptor quiet.
It is. It physically prevents the receptor from going into the nucleus and binding to DNA.
Until the hormone arrives.
Right. The lipid-soluble hormone diffuses into the cell, finds its receptor, and binds to it. That binding event causes the HSP90 bodyguard to let go.
The receptor is now free?
It's free. It usually finds another freed receptor to form a dimer, and then that dimer moves into the nucleus.
And what's its mission in there?
Its mission is to find specific sequences of DNA called hormone response elements, or HREs. It binds directly to the DNA at these spots.
And by doing that, it acts as a transcription factor.
Exactly. It directly alters the rate at which specific genes are transcribed into messenger RNA, either cranking up production or shutting it down.
And the drug effect doesn't happen until that mRNA is translated into a new protein, and that protein builds up. That explains the incredibly long time lag.
It does. But the flip side of that slow onset is a very long duration of action. The effects can last for days, long after the original drug molecule is gone, because the new proteins it caused to be made are still there doing their job.
All right, we've gone through the how at the molecular level. Now we need to connect that to what a doctor or a patient actually sees. We need to quantify it. This brings us to the dose-response relationship, the DRC.
So the DRC is really the cornerstone of rational dosing. It's just a graph that shows you the relationship between how much drug you give and how much effect you get.
And we almost always plot the response against the log of the dose, not the dose itself. That gives us that classic S-shaped or sigmoid curve. Why do we use a log scale?
It has a few huge advantages. First, it lets you display a really wide range of doses on one easy-to-read graph. But more importantly, it linearizes the middle part of the curve.
The part between about 30 and 70% of the maximal effect.
Right. And that straight line portion is the most biologically responsive and predictable range, so it's perfect for comparing drugs.
The DRC lets us talk about two of the most important and most confused concepts in all of pharmacology: potency and efficacy.
I agree completely. If our listener takes away one thing from this deep dive, it should be the difference between these two terms. They are not the same.
Okay, let's define potency first.
Potency is all about the concentration. It refers to the dose of a drug that you need to get a certain amount of response. We typically measure it with the EC50.
The effective concentration to get 50% of the maximum effect.
Exactly. So if drug A gives you a 50% response at 10 milligrams, and drug B needs 100 milligrams to do the same thing, drug A is 10 times more potent.
It's more potent, but is it necessarily better?
Not at all. And that's the key mistake people make. Potency just reflects things like affinity for the receptor. A drug can be incredibly potent, working at microscopic doses, but still be clinically useless if its maximum effect is too low.
And that maximum effect is efficacy.
That's efficacy, or Emax. Efficacy is the true index of a drug's effectiveness. It's the absolute ceiling of the dose-response curve. It's the maximum possible response that a drug can produce, no matter how high you push the dose.
Let's use the classic painkiller example. Morphine versus aspirin.
Perfect. Morphine has a very high efficacy. It can relieve severe, agonizing pain. Aspirin has a much lower efficacy. No matter how much aspirin you take, you will never, ever achieve the same level of pain relief as morphine.
So morphine is more efficacious.
Much more. In the clinic, efficacy is almost always the more important factor in choosing a drug. You need a drug that can actually solve the problem.
What about the shape of the curve itself, the slope? What does a steep DRC tell you?
A steep curve is a warning sign. It means that a very small change in dose can lead to a very large change in response.
So there's not much room for error. The therapeutic window is narrow.
Exactly. It's harder to individualize the dose.
Yeah.
A flat curve, on the other hand, is much safer. I mean, you can vary the dose quite a bit without seeing huge, unpredictable jumps in the effect.
When we take these concepts of potency and efficacy, and then we add in all the real-world patient factors, absorption, metabolism, the disease itself, we get to the ultimate measure, which is therapeutic efficacy.
Therapeutic efficacy is just, does it work in a real patient for a real disease? It's the composite outcome. The sources use the Parkinson's disease example.
Right. An older drug might have looked really potent in a lab test.
But the combination of levodopa and carbidopa is just vastly more effective at actually controlling the symptoms of the disease in a person. The real-world outcome is all that matters.
And a huge part of that outcome is safety, which brings us to drug selectivity.
Right. No drug has only one effect. Selectivity is a measure of how good a drug is at doing the one thing you want it to do, without doing a bunch of other things you don't want.
It's the gap between the dose that helps and the dose that harms.
It is, and it's governed by two things. First, does the drug hit only one type of receptor or target? And second, how widely is that target distributed throughout the body?
Omeprazole comes up again here as a great example.
It's a perfect example of high selectivity. Its target, the proton pump, is almost exclusively found in one type of cell in the stomach, so the drug's effects are highly localized and specific.
And we can quantify this safety margin with the therapeutic index, or TI.
The TI is a formal ratio. It's the median lethal dose, the LD50, divided by the median effective dose, the ED50.
The LD50 being the dose that's lethal in 50% of test subjects, and the ED50 being the dose that's effective in 50%.
Right. You want that ratio to be as large as possible. A high TI means a wide margin of safety. There's a big gap between the dose that works and the dose that's dangerous.
But ultimately, the decision to use a drug comes down to something less mathematical, the risk-benefit ratio.
Always. It's a clinical judgment. Is the potential benefit from this drug worth the potential harm, the cost, the inconvenience? For a deadly cancer, you'll accept a drug with a terrible TI because the alternative is worse. For a mild headache, you wouldn't. It's all about context. #TAG-V, drug synergism and antagonism.
Okay, let's move into the reality of modern medicine, where patients are often taking multiple drugs. What happens when we combine them? Let's start with synergism, when they work together.
Synergism is when the combined effect of two drugs is greater than their individual effects. It's a very powerful tool. The simplest form is an additive effect.
Which is just simple math. The effect of A plus B equals the effect of A plus the effect of B.
Exactly. This usually happens when the two drugs work by a very similar mechanism. Combining aspirin and paracetamol for pain is a good example, or using two different anesthetic gases together, like nitrous oxide and halothane.
But then there's the more powerful version, the superadditive effect, or potentiation.
This is where the magic happens. Here, the effect of A plus B is greater than the sum of their individual effects. 1 + 1 = 3.
How does that work at a molecular level?
It often involves the drugs acting at different points in the same pathway. A classic example is the antibiotic combination of trimethoprim and sulfamethoxazole.
They block two different steps in making folic acid in bacteria.
Right. By hitting the pathway at two separate points, you get a much more profound level of inhibition than you would by just doubling the dose of either drug alone. It's a much more efficient way to shut the system down.
Now for the other side of that coin. Antagonism. One drug gets in the way of another.
Right. The effect of A plus B is less than the effect of A plus the effect of B. This can be a dangerous drug interaction, but it can also be the basis for life-saving antidotes. It all depends on how they interfere with each other.
Let's start with the simplest, physical antagonism.
This is based on pure physics and chemistry, not biology. The textbook example is giving activated charcoal for an oral drug overdose.
The charcoal just acts like a sponge.
It does. It has a massive surface area, and it physically absorbs the drug molecules in the stomach and gut, preventing them from ever being absorbed into the bloodstream.
Okay, next up is chemical antagonism.
Here, the two drugs actually react with each other chemically to form a new, inactive product. Using a chelating agent to treat heavy metal poisoning is the classic case.
The chelator literally grabs onto the lead or mercury ions.
It forms a tight chemical bond, creating a stable, non-toxic complex that the body can then safely excrete.
The third type is fascinating: physiological or functional antagonism.
This is where two drugs have completely opposite effects on the body, but they do it by acting on completely different targets. They never even interact with each other directly.
They're fighting on different battlefields, but for control of the same territory.
That's a great way to put it. The classic example is histamine versus adrenaline on the airways.
Histamine, from an allergic reaction, constricts the bronchioles.
Right, by acting on histamine receptors. Adrenaline, on the other hand, acts on beta-adrenergic receptors and causes profound bronchodilation. The drugs never touch each other's receptors, but their end effects on the same physiological function are diametrically opposed.
And that brings us to the most important type for pharmacology: receptor antagonism. Here, the battle is for the exact same molecular target.
Yes, and we can see the results of this battle play out on the dose-response curve. Let's start with competitive antagonism, the equilibrium type.
So this is our molecular foot race again. The antagonist looks like the agonist, they're fighting for the same active site, and the binding is reversible.
All correct. And because the binding is reversible, the antagonism is surmountable.
Meaning if you add enough agonist, you can eventually win the competition and kick the antagonist out.
You can. So how will that look on the dose-response curve?
Well, you'll need more agonists to get the same effect, so the curve will shift to the right. But since you can eventually win, you should still be able to reach the same maximum effect.
Precisely. You get a parallel rightward shift of the DRC. Emax is unchanged. The classic examples are atropine blocking acetylcholine or naloxone blocking morphine.
Now let's contrast that with non-competitive antagonism. We're back to the allosteric side door site.
We are. The antagonist binds to that separate site and deforms the receptor. The agonist might still be able to bind, but the receptor is broken. It can't signal properly.
And because the receptor itself is sabotaged, adding more agonist is useless. The antagonism is unsurmountable.
It is, which means you can never reach the original maximum effect. Non-competitive antagonism causes a flattening of the DRC. The Emax is suppressed. A certain fraction of your receptors are just taken out of the game completely.
And finally, there's a special case called non-equilibrium antagonism.
This one looks like non-competitive antagonism on the DRC, but the mechanism is different. Here, the antagonist binds irreversibly to the receptor.
Often with one of those covalent bonds we talked about.
Exactly. It might bind at the active site, like a competitive antagonist, but because it never lets go, the law of mass action doesn't apply. The agonist cannot displace it.
So it's effectively unsurmountable, just like the non-competitive type.
Precisely. The end result is the same. The DRC is shifted, right, and the Emax is flattened. The only way the body can recover is to physically synthesize brand new receptors. Phenoxybenzamine, an alpha blocker, is the textbook example of this.
That was an incredible tour through the landscape of drug action. We started with the basic mechanics of enzyme inhibition, seeing how one type shifts the KOM while the other reduces Vmax.
And we built all the way up to the incredible complexity of the 5 major receptor superfamilies. We contrasted the millisecond speed of an ion channel with the hours or days required for a nuclear receptor to actually change gene expression.
We really nailed down those core ideas of affinity, how well a drug binds, and efficacy, its ability to actually produce an effect.
Yeah, and we saw how looking at a dose-response curve, its slope and its maximum, can tell you so much about a drug's safety and its clinical potential.
Understanding these mechanisms really is the foundation of rational medicine.
It is. It explains everything. Why a partial agonist like buprenorphine is a safer option, why a steroid cream keeps working long after you've washed it off, why some drug combinations are synergistic.
Our sources make it clear that a receptor has 2 fundamental jobs: recognize the ligand and then transduce the signal. And for a long time, we've thought about that in simple on or off terms. An agonist has an intrinsic activity of 1.
But the reality of those G protein systems, Gs, GGQ, and others, it hints at something much more sophisticated. We now know that the precise way a drug makes a receptor change its shape can determine which of those several G proteins it prefers to couple with.
Which brings us to a final, really provocative thought for you to consider. What if the future of drug design isn't just about finding drugs with the highest affinity or about getting that intrinsic activity to a perfect 1.0?
What if it's about designing a ligand that is a skilled driver, a drug that doesn't just turn the key, but specifically steers the receptor's downstream signaling toward one desired pathway, like the GQ pathway, while completely ignoring another, like the G pathway, that might cause side effects?
That's the concept of functional selectivity, or a biased agonism.
It is. It goes so far beyond the simple on-off switch model. It suggests we could create drugs that are not just receptor-specific, but pathway-specific. Drugs that can pick and choose the exact cellular response they want to create. That is the next frontier of pharmacology, and it's something well worth exploring further.